All right, so let's go back to the questions from last time. First, we had what type of microbe is the causative agent of ergotism, and there we're talking about Claviceps purpurea, and it's a fungal microbe. Uh, Two, true false. Ergotism is a disease caused by ingestion of either ergotamine or lysergic acid diethylamide. This is a false statement. The reason it's false is because ergotism is caused by ingestion of ergotamine only. And so when you see a statement like this, and within one statement is something that is both true and something that is false, the one false part negates the entire thing. So you end up having to say the whole thing is false, um, even though it's part true, but altogether it's all false. Okay, so three, how is epidemiology helpful in understanding and treating a disease like ergotism? Well, when it comes to treating disease, one of the important things is to see the bigger picture and to have a pattern for the disease. It's one thing to say that someone is hallucinating, and it's another thing to say that they're hallucinating and it's a common symptom in one town and it's fall and everyone had been eating rye. And as you build up all of those different patterns that you get from epidemiology, you're able to come up to a better diagnosis. And if you have a better diagnosis, then you're going to have better treatments. Okay, next we're going to look at this second example of disease. And in this case, we're going to be looking at an organism called Pseudomonas syringae. You can see in this picture some of the bacterial cells of this species sitting on the leaf of a plant, with the plant leaf colored in green here. And we also have the statement at the top, all pathogenicity has specificity. And that's going to be an important phrase to remember, because whenever we're talking about pathogens, we're going to be trying to think about the range of pathogenicity. And so there's always going to be some level of specificity. Some pathogens will be highly specific, only targeting one particular organism. Other pathogens are more generalist. Just like last time, I'm going to show you guys some questions before we get into the material, so you can start thinking ahead about what sort of questions you might be able to dig out of this material. So take a quick look at these. As a human, should you be concerned about getting sick from a pathogen like Pseudomonas syringae? Why or why not? True-false question. E- strains can be used to prevent frost damage. And three, draw and explain the molecular mechanism for how ENA proteins promote ice nucleation. Okay, so those are some of the concepts that we're going to be looking for. Okay, so this organism is Pseudomonas syringae. If you look at the name, it actually tells you a little bit about the organism, so it can help whenever you're looking at microbial species names to start recognizing things that pop up a lot and what those roots to the word indicate. So if you see the term pseudo, that means not genuine. Okay, so that means that somebody saw something in this microbe that would change sometimes. And when they saw it changing, they said, aha, you are not genuine. You're not always showing me the same exact phenotype, so you're going to be called a pseudo. A monad or a monas is referring to singular, so you don't see the cells in groups of 2 or 4 or larger groups. You typically see them isolated from each other. And then syringae, as you might guess from that term sounding like syringe, refers to a tube. And in this case, it's a little bit confusing. People get this mixed up quite a bit. The syringae refers to the first plant that it was isolated from, which had these very narrow tube-like branches. But it turns out that Pseudomonas syringae, the bacterium, makes a needle-like structure as part of its infection mechanism. So people were like predicting ahead that syringe would be an important name for this organism. I'm also going to suggest that if you want to learn microbiology, you're going to want to maintain a list of all the species that you come across. So as you're reading, as you're watching videos, as you're doing research on your own, just find someplace in your notebook, have the left side be genus and species names, have the right side be key traits. Okay, and so if you haven't started that yet, it's a good one to start with is Pseudomonas syringae, and as you write down that name, uh, we'll get in the next couple slides here some key traits that you can associate with Pseudomonas syringae that would be true of that organism, and the key to it being a key trait is you want it to be not true of something else. We'll come back to it later where sometimes it's easy to say something like causes disease. That's a little bit too vague. We want to get more specific than that. Okay, so let's dig into the details on this organism. It's an opportunistic plant pathogen. It can infect plants. As an opportunistic plant pathogen, it can infect plants, but it cannot breach the plant cell wall. So if it happens to land on the surface of a healthy plant, it's not able to be pathogenic. It requires a wound for entry. And so that wound to the plant is the opportunity. So that's where the term opportunistic comes from. It means not always pathogenic. There has to be some sort of starting conditions that are just right. Okay, so Pseudomonas syringae has some pretty cool abilities. It's able to promote ice nucleation and raindrop formation in the upper atmosphere. You can actually isolate this organism from the center of snowflakes or hail. It's easier from hail. So, if by chance there's one of those days when hail is coming down and you can get balls of hail that are, a couple centimeters in diameter, and if you take a handful of those balls of hail and you bring them into the lab and then you freeze them and you very carefully cut them in half under sterile conditions so that you're not introducing any new microbes, you can sequence for DNA in the center of the hailstone and you'll find the DNA of Pseudomonas syringae in there. That's pretty amazing that that's something that you can find inside of hail. Okay, so there's a little bit of atmospheric knowledge going on here. So in the upper atmosphere, water remains liquid way, way, way below where we would think of water freezing and becoming a solid. We think of it becoming a solid at 0 degrees Celsius, but in the upper atmosphere, the pressure is so low that it'll actually stay as a liquid down to minus 40 degrees Celsius. It turns out that that's a really difficult temperature for our planet Earth to reach. So it's really hard for liquid water to condense into snow or even into raindrops. So the presence of Pseudomonas syringae actually helps increase condensation of water by changing the freezing point. It shifts it from minus 40 to a freezing point of minus 4 to minus 2 degrees. Okay, so now let's think about the impact of these traits. Pseudomonas syringae, is it a good bacterium or a bad bacterium? We've heard that it can be a plant pathogen, and we've heard that it can cause water to fall. So there's an interesting dynamic here where this organism can make it rain, and by making it rain can encourage plants to grow, but then it can also cause frost formation, which can then damage and weaken or even kill a plant if the damage is severe enough. So this organism can both help plants grow and it can, in some cases, be lethal towards plants. So it's hard to figure out exactly where to place it. It gets complicated. There's some really fascinating studies showing that you can have an ecosystem where, almost all of the conditions weather-wise are exactly the same. In one spot you've got desert, in another spot you've got plants growing, and there's a cycle that forms where Pseudomonas syringae from the plants will get mixed up, they'll dry out, they become dust, they go up into the atmosphere, they make it rain, and then it rains, and then you make more plants, which allows there to be more Pseudomonas syringae, and so you get these positive feedback loops that allows there to be growth in areas where otherwise there wouldn't be, and it's a combination of the bacteria and plants interacting with each other. Okay, so let's get into a couple of questions about how and why. So the question of why is going to be, why would Pseudomonas syringae want to nucleate ice? What's the benefit for this little bacterium? Well, the frost damage to plant leaves is going to release nutrients, and those nutrients feed the bacteria that allows the bacteria to grow, and the frost damage provides an opportunity for infection. The rain-snow condensation provides a mechanism of transport for the organism. You can imagine that if Pseudomonas syringae, it dries out in a dry season, and it becomes all kind of dusty, and it gets flung way up into the upper atmosphere, and it's hanging out in the upper atmosphere. If it didn't condense raindrops, it could just hang out there forever or get cast out into outer space. But by being able to condense water around it, it sort of builds a parachute for itself that allows it to fall gently back down to Earth. And in many cases, rather than actually landing on the soil, the raindrops are going to land onto a new plant leaf, which is going to be a new potential opportunity for the Pseudomonas syringae. If we look into the question of how does Pseudomonas syringae nucleate ice, this is going to get into the proteins and the molecular mechanism that is the basis for this process. It works like this. They express a set of proteins outside the cell called ice nucleation activity, or INA for short, INA proteins. These proteins outside the cell have a specific repeating structure. You can see in the image here that this would be present on the outer surface of our bacterial cells. The green arrows are showing you that there's beta sheets. You can see that the beta sheets have multiple repeats, so they sort of form this long tube structure of beta sheets. And then at the top of every beta sheet, this image is just showing off one particular amino acid. There's amino acids residues all along the chain of the protein here, but we're just highlighting one to show you that at the tips of these beta sheet curls, there's going to be this repeating amino acid. It might already look familiar. So this is a tyrosine side group, and tyrosine has the benzene ring with a hydroxyl pointing out away from the peptide backbone. So we've got this hydroxyl pointing out. The way that these tyrosine hydroxyls are organized within the overall structure of the protein is that all of these tyrosine residues are pointed out in basically the same direction. So these cells express these proteins outside the cell. It has in the protein a hydrophilic N-terminus and a hydrophobic C-terminus. It's the hydrophilic or water-loving N-terminus that's exposed outside the cell, and it's the hydrophobic C-terminus that stays anchored in the membrane. And then we have this highly repetitive beta sheet structure with conserved tyrosine residues. And all of those hydroxyl groups pointing out from tyrosine, each one of those can promote hydrogen bonding with water. And as they promote hydrogen bonding with water, it's going to hold a water molecule in place. And for each water molecule held in place, that's going to make it easier for water to hold the next water molecule in place with even more hydrogen bonds. So if you're thinking back to how ice forms and the hydrogen bonding that happens with water, this protein can mimic water in a crystal state, and it acts as kind of like a seed crystal to get water more likely to hold that rigid hydrogen bond-based shape of solid water, unlike the transient hydrogen bonds of liquid water. Okay, next up is, knowing all of this, can we make use of it? And it turns out there's a host of interesting things you can do once you understand what this microbe is doing. First of all, you could take ENA minus or ice minus strains. So these are strains that don't express the ice nucleation protein outside the cell. And if you coat plants in these ENA minus strains, it prevents the ENA plus strains from having an ecological niche to grow on. So it stops frost damage from happening. It's a pretty amazing trait that you can just take away this one protein, and then they lose the ability to cause frost damage, and you can save your plants from a freeze break that happens in spring. The next thing, for anyone who skis or snowboards, if you go out to one of the ski lodges and you look around, you might notice that sometimes you'll see them making snow. Now, a lot of times people will look at that and assume that, it's just mixing water and air together and you get snow. But if you've ever made ice cubes in your freezer, you know that you can't just mix water, you can't just make water cold and turn it into snow. It doesn't work that way. Water wants to turn into ice, but you can encourage water to turn into snow if you mix dead Pseudomonas syringae in to nucleate the snow. It could be living Pseudomonas syringae too, but having live cultures around is not really practical. You can actually freeze dry Pseudomonas syringae, kill it, and the proteins maintain their ice nucleating ability, and so they'll be in this kind of like powdery mixture, and so that mixture of water and cold and the powder from Pseudomonas syringae gives you snow. And it's kind of amazing that we can keep snow going as long as the temperature is good and you've got water in these machines. Okay, finally, you can actually release Pseudomonas syringae from planes, and if there's enough water vapor in the atmosphere, the presence of increased Pseudomonas syringae can encourage rainfall. So there's all these things that you can do with both protecting plants, making snow, encouraging rainfall, once you understand that there's this invisible microbe at the heart of this really important process to planet Earth. Okay, so let's go back to these questions and see how you do. Question 1. As a human, should you be concerned about getting sick from a pathogen like Pseudomonas syringae? Why or why not? 2. True false. E- strains can be used to prevent frost damage. 3. Draw and explain the molecular mechanism for how ENA proteins promote ice nucleation. Okay, we'll talk about the answers to these questions next time.